Okay, well, I'm going to get started with a couple of background notes about SGP. I think a lot of people here are quite familiar with SGP, but for those of you who aren't, it's just reading from our website, an open community-oriented research consortium that tries to address questions of environmental evolution across Earth history through statistical analyses of the sedimentary geochemical record. It is a research consortium kind of styled after research consortia in the biomedical sciences. So there's greater than 150 collaborators around the world that are involved. And we have kind of two aspects of our project. One is aggregating sedimentary geochemical data. And the more important part of it is trying to analyze that sedimentary geochemical data through time. Of course, the first thing that we want to know if we want to be able to analyze sedimentary geochemical data through time is what these proxies actually tell us. And so in order to help us all better understand all of the different various proxies, we've started this Proxy Primer series. And the goals of this are to accessibly explain the basics of a geochemical proxy or method to a broad audience, to create a proxy video library that people can go back to as they want to learn more about proxy, and finally, to provide an open space for interaction with the experts on each proxy. So today, after Maya is done, we'll be able to turn off the video recording and ask her questions. Just a quick reminder of our 2022 schedule. We had a proxy primer with Zunli Liu on iodine calcium. Today, we're hearing from Dr. Gomes on sulfur isotopes. October 11th, we have an in-person meeting, update meeting and social hour at the GSA meeting. And then on November 16th, Dr. Selva Marroquin from Caltech will be telling us about carbon isotope chemostratigraphy. So with that, I will turn it over to Dr. Gomes. I'm very excited that she's here to meet with us. She did her undergrad degree at Wesleyan University before moving on to Northwestern to do her PhD. She then did her postdoc, at Harvard, which is where I met her, and Washington University in St. Louis. She then moved on to a faculty position at Johns Hopkins, where she is now. And through all of this, she's become one of the world's experts in sulfur isotopes, which are, of course, one of the main ways that we know about the ancient Earth system. And so I'm very excited to have a proxy primer on that. She's worked all over the world, both in locations and time. So she's worked in modern lakes and oceans. She's worked on microbial mats, and she's worked in the geological record. So with that, Maya, please take it away.
Great, thank you, Eric. So thank you for that lovely introduction, and also thank you and the whole SGP team for all that you've done to create this community and resource. I think it's really a wonderful thing for geochemists, geologists, and paleontologists, and I'm really happy to be able to be a part of it by giving you this proxy primer today. So today I get to do what I love to do, which is talk about sulfur isotopes and how we can use them to understand various events in Earth history, but then also some of the issues that we might come into when we're trying to either sample for them, measure them, or interpret them. Okay, I'm gonna get my laser pointer working, and I'm advancing for my first slide. So if you don't see it, please let me know. So this first slide gives us some motivation. And so this is arguably one of my favorite plots in all of science. This is a sulfur isotope record of sedimentary pyrite from about 3.7 billion years ago until the present. And what you'll see is this is a really, really long and complete record. So it's one of our longest and most complete geochemical records. But the other thing that you can see is that there's actual structure in there. And this sulfur isotope record tells us about the coevolution of life in the Earth system. So I'm going to color code the different symbols to draw your eyes to some different patterns, as well as put some lines on this plot to help us interpret it. So the top line is our best reconstruction of the sulfur isotope evolution of the marine sulfate reservoir. And then the lower line is offset by about 50 per mil, which gives you a sense of what a range of 50 per mil is in sulfur isotope space on this plot. And so what you can see is that there's differences in the range of sulfur isotope values of sedimentary pyrite throughout Earth history. Prior to the Great Oxidation event, the range was very, very low, likely because the green sulfate levels were low. But then once oxygen rose in our atmosphere and there were significant rates of pyrite weathering on land, oxidative pyrite weathering on land, then marine sulfate levels increased. And so you're able to preserve a larger range of sulfur isotope compositions of pyrite. And we'll talk about why that is later. But I just want to explain this first. Then close to 0.7, 0.8 billion years ago, we see another increase in the range of sulfur isotope compositions of sedimentary pyrite. And this is sort of temporally linked with what some of us might think about in terms of another stepwise increase in oxygenation. So perhaps a neoproterozoic oxygenation event at this time. So work is still being done to sort of understand this transition. But the point I wanted to make here is that this is a really robust record. So you might be coming to SGP because you want to analyze this record and do some sort of statistical analysis of it to learn about Earth history. Or maybe you're thinking about just going out to a section and measuring a strat column and then sampling it in order to look at sulfur isotope records in context of other geochemical records, like for an example, an organic carbon record. And so this is a really fun thing to do because you can do these reconstructions and then interpret your geochemistry in terms of the sedimentary in terms of the sedimentology and tell some interesting or make some interesting interpretations about environmental change in the past. And so this is an example that's near and dear to my heart. There's a lot of different examples, but this is a sulfur isotope record from a Cretaceous oceanic anoxic event 2, which is a period of widespread organic carbon burial in the Cretaceous. That's temporally linked with large igneous province volcanism as well as extinctions. And so this study was examining how the sulfur cycle changed through this event and promoted enhanced primary productivity during the event, and then ultimately a drawdown of sulfate, decreasing sulfate levels and returning the system to pre-excursion conditions. So again, I'm glossing over a lot of the details here, but these records can be really valuable, and we like to measure these records because the sulfur cycle is intimately linked with the carbon and oxygen cycles. And so in order to understand the history of our planet, one thing that we do is try to reconstruct atmospheric oxygen levels and atmospheric carbon dioxide levels through time. So we want to know about atmospheric carbon dioxide levels, of course, because that impacts climate. And we want to know about oxygen levels because oxygen levels impact which organisms can live in different environments. And so in order to model this, we need to consider different reactions that impact these atmospheric gases. And so one of the ones that a lot of us think about is photosynthesis, which is the process by which organisms can use CO2 and water to produce organic carbon and oxygen. And this can have a huge impact on atmospheric CO2 and oxygen levels. And that's because the burial of organic carbon constitutes the biological pump that removes CO2 from the atmosphere. So you're taking CO2, reducing it, and then burying it in sediment. So that's the way that it gets out of the atmosphere. This is also associated with the net addition of oxygen into the ocean atmosphere system because you're leaving that oxygen in surface environments. So this reaction is incredibly important. However, only a very small portion of that organic carbon that's actually produced in surface waters gets buried. There's a whole lot of reactions that consume that organic matter and prevent that organic carbon from getting in the sediment and being buried. And one of the most common ones and ones that we'd love to think about is oxic respiration. And so this is what we do and what most animals do. And so they use oxygen to consume that organic carbon, producing CO2 and hydrogen, and water. And so in certain places, if you have high rates of primary productivity and that drives this reaction forward, or if you have slow rates of transport into your system, then you can run out of oxygen. And then you form something like an oxygen minimum zone environment. But in absence of oxygen, there's a whole lot of other compounds that can be used to consume organic matter. And so this is what we think of as the biogeochemical ladder, where we see different reactions consuming organic matter in space and time in some cases. And ultimately this order is due to how much energy you can get from the reaction. So usually oxic respiration happens first because you get a lot of energy from that. And then you go through all these other oxidants. But sulfate reduction is an incredibly important process because the marine sulfate reservoir is the largest oxidant pool in the modern ocean. And so therefore, up to 50% of organic carbon degradation in marine sediments occurs through sulfate reduction. So we can just think about the concentrations here, right? So if, some saturated seawater might have something like 350 micromoles of dissolved oxygen. And, if you're in a really nitrate-rich environment in the ocean, there might be 45 micromolars. But sulfate concentrations in the modern ocean are 28 millimolar. So that's orders of magnitude more. And so that's why the sulfur cycle is such an important part of biogeochemical cycling. So the reaction that we think about is microbial sulfate reduction, where organisms use sulfate to consume organic carbon, producing bicarbonate and hydrogen sulfide. And that hydrogen sulfide is important for many reasons, but I'm just going to highlight a few. One is that hydrogen sulfide is toxic to most eukaryotic organisms. So if that hydrogen sulfide diffuses into the water column or accumulates in the water column, then that can impact organisms. It can lead to extinctions. The other reason why it's important is hydrogen sulfide is a powerful reductant, and so it can also consume oxygen and impact oxygen levels in bottom waters. And also, that hydrogen sulfide can react with iron to form pyrite, which is buried in the sediment, or with organic matter to form sulfurized organic matter. And both these fluxes of reduced sulfur phases, so this burial of reduced sulfur phases, represents a net addition of oxygen to the atmosphere. You know, these are all redox reactions, right? So we're taking CO2, putting some electrons on it, and now we're forming organic matter. And then that organic matter, we take the electrons from that and then put it onto hydrogen sulfide. And so we're just moving this reducing power into the sediment. And that represents a net addition of oxygen to the atmosphere if that sulfate comes from marine weathering of sulfate minerals on land. So, but this reaction has a different impact on on the carbon cycle, because instead of that carbon being buried in the sediment, that carbon is remineralized, and that bicarbonate can rejoin the dissolved inorganic carbon reservoir either in the water column or in the sediment pore waters. So in order to reconstruct oxygen and carbon dioxide levels in our atmosphere through time, we really need to consider all these different cycles. And we can use sulfur isotopes in combination with other proxies to try to reconstruct these processes. And specifically, the way that we do this is considering a box model. And so this is just an example, but we use box models to model the evolution of the marine sulfate reservoir, both in terms of its concentration and isotopic composition, because that concentration and isotopic composition will vary in response to changes in the magnitude and the isotopic composition of the inputs and the outputs. And so this sort of box model interpretation comes up with specific predictions that we use to try to extract paleoenvironmental information from our records. So specifically, when we see increases in sulfur isotope composition of sulfate that is preserved in the geological record, we usually take that to mean there's enhanced pyrite burial, perhaps in low oxygen systems, a change in the sulfur isotope fractionation associated with sulfate reduction that might be due to changes in organic matter or something else like that. Whereas a decrease in the sulfur isotope value, we usually interpret as being due to increased rates of weathering, volcanism, or decreased rates of pyrite burial, perhaps because there's less low oxygen systems in the ocean. And then we just assume that the pyrite follows along, but is offset by that sulfur isotope fractionation value. And again, I'll go into a lot more specifics about these different processes and why we make these interpretations. But what I wanted to talk about this early on, just so that you can see the power in using sulfur isotope records. But I have to make a really important caveat. And that's that we can do this if and only if we have used careful tools, so petrological, geochemical, and stratigraphic tools, to determine that these sedimentary sulfur isotope records are actually recording secular variation in seawater conditions. And so I need to make this caveat, right? So I love sulfur isotopes. I've been lucky enough to spend much of my career studying sulfur isotopes, but I have to accept, and we all have to accept, that they can be reset and that they can be altered. And sometimes that alteration actually gives you some information about some really interesting environmental change. But in order to actually interpret these records as recording information about ancient biogeochemical cycling, we really need to prove that these signatures are actually recording secular variation in seawater conditions, because that is what we're trying to get at. Okay, so this caveat needs to be made, and I'm going to talk to you a lot about how you can assess your sulfur isotope records and try to infer what sort of information they're recording, and then how you make those interpretations. Okay, so let's start with the basics. So I'm an isotope geochemist because I love isotopes. So they're forms of the same element that have a different mass. So because they're the same element, they can participate in all the same reactions, but because they have a different mass, that means that the rate of those reactions might differ. And so by measuring the isotopic composition of reactants and products, we can learn something about those reactions, or perhaps mixing of different reservoirs. So what we're talking about today are sulfur isotopes. And so there are 4 stable isotopes of sulfur. We most commonly measure sulfur 34 and sulfur 32, which are the most abundant ones. It is possible to measure 33 and 36, and that could give you extra information. But Eric and I agree that that should be a subject of a different SGP Proxy Primer. And today we're going to really concentrate on the major isotopes, so 34 and 32. But I do want to point out that if you're thinking about measuring minor sulfur isotopes, you should always measure the more abundant sulfur isotopes first, just so that you know what variability you have in your system and can come up with hypotheses. Because the measurement of 33 and 36 are labor and time intensive, and you want to make sure that you have a really good question to ask before you sink a lot of time into that. Okay, so we quantify sulfur isotopes as the ratio of sulfur 34, just 32 in your sample, normalized to a standard. It is a really small value, and so we multiply it by 1,000, so it's a number that we can think about. And we express it in per mil units as a deviation out of 1,000. So percent is deviation out of 100, per mil is deviation out of 1,000. And the standard is the Vienna Canyon Diablo troilite, which is an iron sulfide mineral from within the Canyon Diablo meteorite, which formed meteor crater in Arizona. And so it's a really great standard. And so I always find it whenever I go to a museum and take a picture of it. So this is me at the Smithsonian, being really happy that I'm at the sulfur isotope standard. So the primary process that impacts sulfur isotope values in low temperature environments is microbial sulfate reduction, which is this reaction that I showed you earlier, which is responsible for up to 50% of organic carbon degradation in modern sediments. During this process, organisms preferentially utilize the light isotope of sulfur such that the hydrogen sulfide that's produced is depleted in the heavy isotope, or we say it has low sulfur isotope values. Or if we're talking among friends, we might say it has light sulfur isotope values. The residual sulfate is enriched in the heavy isotope, and we say it has high sulfur isotope values. Or when we're talking among friends, we might say it is heavy. So we can quantify the sulfur isotope fractionation associated with sulfate production using this epsilon value, which is just the sulfur isotope difference between sulfate and sulfide. And this value can be up to 70 per mil. And so I have this plot from Sim et al. Because this is a really important paper in sulfur isotope geochemistry, because this really represented a change in the way that we interpreted sulfur isotopes. Prior to the publication of this paper, the only reported, or the maximum sulfur isotope fractionation associated with sulfate reduction that was reported from lab studies was 46 per mil. And so it was thought that in order to get sulfur isotope values higher than 46 per mil, you had to invoke additional processes. And so this is a schematic to show you what those processes were. And so specifically Canfield and Thandrup introduced the idea that sulfur disproportionation reactions would increase the sulfur isotope difference between sulfate and sulfide. So disproportionation reactions are reactions where some sort of intermediate species is both reduced and oxidized as part of the reaction. So here, zero valent sulfur is both oxidized to sulfate and hydrogen sulfide. And there's a sulfur isotope effect such that the hydrogen sulfide gets lower. So if you go through repeated processes of reoxidation and disproportionation, then you can eventually generate a sulfur isotope offset between sulfate and sulfide that's greater than 46 per mil. But this was all changed once the Sim paper was published, which showed that you can get fractionations of up to 70 per mil for microbial sulfate reduction alone. So I want you to think about this when you're doing literature searches, because if you look through the literature prior to 2011, you'll commonly see people interpreting sulfur isotope offsets between sulfate and pyrite that are greater than 46 per mil as being due to sulfur disproportionation. And so when you read those interpretations now, you should think about that with a grain of salt, because this is no longer a unique interpretation. Okay, so I said that sulfate reduction is the main process that impacts sulfur isotope patterns. I also want to mention that sulfide oxidation reactions happen. So in the modern ocean, up to 90% of the hydrogen sulfide that's produced by microbial sulfate reduction is reoxidized. So do we have to worry about these oxidation processes? And usually we don't really consider them, and that's because the sulfur isotope fractionation associated with these processes are really small compared to sulfate reduction. This is a paper from 2019 where they showed that you could get really, really large fractionations at high pH. And this is in part due to equilibrium isotope fractionation associated with speciation of hydrogen sulfide at high pH. But at marine pHs, generally the sulfur isotope fractionation associated with sulfide oxidation is small. And we really consider sulfate reduction to be the main lever. And studies have also shown that a major portion of the sulfide oxidation that occurs in marine sediments actually is sulfate that's leaking back out through the sulfate reduction pathway. So I do think it is pretty robust to consider sulfate reduction and the reversibility of that sulfate reduction pathway as a main factor in interpreting sulfur isotopes in marine sediments or in sulfate reduction systems. Okay, so I told you that the sulfur isotope fractionation can be up to 70 per mil. What controls the magnitude of that fractionation? So when you think about sulfur isotope fractionation, I want you to think about two things. One is sulfate reduction rates and the other is sulfate levels. So let's start with sulfate reduction rates. So it's been known since the 50s that there's a negative relationship between sulfate reduction rate and fractionation. Higher rates of sulfate reduction usually tend to favor lower fractionations and vice versa. Now, there's been a lot of work done on this since the 50s to refine this relationship and think about how each step of the sulfate reduction pathway and the fractionation associated with that step impact the magnitude of the fractionation, as well as the reversibility of the reaction will also impact the magnitude that you see. And so there's a whole series of literature that you can look at, that you can read about this, and I encourage you to do so. These are some really great papers. But for the purposes of this, I just want you guys to think about what impact this rate dependence trend would have on sulfur isotope compositions of sulfide in the zone of sulfate reduction. And so I have a schematic here which shows sulfur isotope compositions of sulfide with depth in a zone of sulfate reduction. So this could be a marine sediment, this could be a euxinic water column, this could be a microbial mat. Pick whatever your favorite system is. And so given this rate dependence trend, we can come up with a prediction of what we would see for sulfur isotope compositions with depth in this system. So at the surface, at the top of the zone of sulfate reduction is usually where we measure the highest rates of sulfate reduction and or sulfide oxidation. And because the highest rates of sulfate reduction occur here, that means that we have the lowest fractionation, which favors high sulfur isotope compositions of sulfide. I know it's a little bit confusing, but recall again that this fractionation is the sulfur isotope difference between sulfate and sulfide. And so when we have a low fractionation, then the hydrogen sulfide sulfur isotope value is relatively high because it's close to the sulfate reservoir. In contrast, at depth, where you tend to have lower rates of sulfate reduction, you would have a larger fractionation, which favors the formation of hydrogen sulfide with really low values. And so these are the trends that we would expect to see if this is what is driving sulfur isotope variability in your system. Fun. So the other thing to think about is the reservoir effect, which is described by a Rayleigh fractionation trend in a closed system. So everybody look at this side. I'm sorry about this stuff over here. So this follows what's called the Rayleigh fractionation trend. And so this plot shows you how this works. And so this plot is showing the trajectory of isotope values of a reactant and a product in a irreversible reaction that carries an isotope effect. And it's showing you that isotopic evolution as the fraction of the reservoir of the reactant consumed goes from 0 to 1. So for our purposes, if you're at 0, then that means that all of your sulfur is in the form of sulfate, versus if you're at 1, then all of your sulfur is in the form of sulfide. So as the reaction starts, the instantaneously forming product, what's indicated by this dotted purple line with P prime, that instantaneously forming product is offset from the reactant reservoir by the sulfur isotope fractionation value. But then as the reaction proceeds, the reactant becomes enriched in the heavy isotope. And the instantaneously forming product follows along that trend, but is offset by the fractionation value. Now, we're very rarely able to actually measure the instantaneously forming product. And instead, what we're measuring is a pooled product. So we're just measuring isotope compositions of ambient sulfide. Well, that pooled product, or the ambient sulfide, evolves to this same isotopic composition as the starting sulfate reservoir. And so there's a few important things that come out of this. One is that if your reaction goes to completion, then there'll be no apparent isotope effect, because everything that goes in is what comes out. But the other thing is that this depends on sulfate levels, because if you have lower sulfate levels, then you're more likely to drive the reaction to completion than if you have high sulfate levels. The analogy I like to make is with trail mix. So trail mix is like a collection of nuts and berries and maybe some chocolate chips that you take on a trip. Now, if you have a huge bag of trail mix, then what you'll do is, if you're like me, you'll eat out all of those chocolate chips. And that means that your stomach will be enriched in chocolate chips relative to that bag of trail mix. But if you have a really, really small bag of trail mix, then you might start by eating out those chocolate chips, but eventually you're going to eat the whole thing. And the ratio of peanuts to chocolate chips in your stomach is going to be the same as the bag that you started with. So that's why the size of the sulfate reservoir matters. And this is very specific predictions for what we would see in terms of sulfur isotope composition of pore water sulfide with depth in the zone of sulfate reduction. Specifically, we see that it increases with depth. And this is just following along the trajectory that we predicted for a pooled product. Now, this has been known for a long time. And so this plot that I have the axes up that I wasn't showing you is a paper from 1979 where Jorgensen modeled these processes and showed that closed system sulfur cycling can impact the isotopic trajectory of sulfate in this case. But this study also showed that system openness plays a big role. So if you have an open system where lots of sulfate is diffusing in and hydrogen sulfide is diffusing out, then you won't have the isotopic evolution, or you won't have the reactant reservoir evolving to that high values because you have new addition of sulfate. So closed versus open system dynamics do impact the patterns that you see with depth in the sediment. So I like to think of these two things as end numbers, right? So in reality, natural systems are more complicated than these sort of end members that we like to talk about. But I do think that these are useful in the way that we think about sulfur isotope variability with depth in a system. And the truth of the matter is, looking at the scale that you look at also matters. So it's definitely possible that you could have this differential metabolic trend superimposed on the Rayleigh trend if you're able to look at the top of the zone of sulfate reduction at really fine resolution. So scale also matters in terms of what signature you're capturing and what it might indicate. And so again, these are two end members. In most systems, probably both of these processes are operating. You just have to figure out which one is more important. And so I'm just going to give a little bit of idea of what types of systems these are common in. And again, there's exceptions to every rule, but generally we tend to find the rate dependence trend dominating in systems with high degree of system openness and or high sulfate levels. So some classic examples are these hypersaline microbial mats from Baja, California, or the Black Sea, a euxenic water column. Whereas the reservoir effect is usually more common in systems with a low degree of system openness and low sulfate. And so classic examples are sediment pore waters, but these can also be found in low sulfate microbial mats and euxenic systems. So again, these are just two end members, which I think are helpful to think about, but likely in a lot of systems, there's some overlap. Okay, so unfortunately, we're not able to directly measure hydrogen sulfide and sulfate in the geological record. Instead, we're measuring their archives. And so what are we actually measuring? So we usually measure sulfate as in carbonate associated sulfate. And so this is sulfate that's substituted into the carbonate lattice. And it usually captures the signature of sulfur isotope compositions of marine sulfate in surface environments where carbonates are formed. So if you're lucky enough to work in the Mesozoic or Cenozoic, you might have some pelagic organisms, planktonic organisms that produce calcium carbonate tests, and then that can capture your sulfur isotope signatures in CAS. Or perhaps if you're working earlier in the Earth history, you might want to look for something like bivalves, which again could preserve this really well. That's not a bivalve, that's a brachiopod, I'm sorry. You want to look for brachiopods or something like that. Or other time periods in Earth history, you might be measuring CAS from carbonate textures that might indicate their cements, or something like that. So looking at your sample and understanding what the carbonate phase is can be really important in thinking about what location you're actually capturing information about marine sulfate, as well as how you might interpret that. Now, pyrite is formed in where there's hydrogen sulfide, so usually in sediments, although it can form in the water column. And so it's usually capturing a signature of sulfide at depth. And so this actual capital delta value that we measure in the geological record, which is the sulfur isotope offset between carbonate associated sulfate and pyrite, extracted as CRS, that value is sort of capturing a spatially separated signal. It's usually capturing sulfate in the surface environment and then pyrite at depth. Okay, so now we're going to go into what are these two different pools? What are we actually measuring? What are the things that you want to consider when you're in the lab? And then we're going to sort of back out and think about what you want to think about in the field and potential pitfalls with interpreting these records. So carbonate-associated sulfate, well, I should say both carbonate-associated sulfate and chromium reducible sulfur are operationally defined terms. And so they're sulfur that are extracted by these particular lab methods. And so even though we assume that all CAS is sulfate that's substituted into the carbonate lattice, we're not always verifying that. We're sort of making an assumption that this extraction procedure is only capturing that. So carbonate-associated sulfate is the sulfate that's released from carbonate upon acidification. So what we do is we take our sample, we grind it up, and then we rinse it with various types of solution to remove forms of sulfate that are not in the form of carbonate-associated sulfate. And so what we do is we, there's various types of rinses that people use, but you can use something like sodium chloride, which should get rid of gypsum anhydride, and perhaps organic sulfur. Some people like to use bleach or peroxide to oxidize that organic sulfur, things like that. And so these are various rinses that we use. And the reason for this is that the way that we actually capture that sulfate is we acidify it. All of that sulfate is released from the carbonate lattice, and then we add some barium chloride, which reacts with that sulfide before forming barium sulfate, which is a highly insoluble mineral. And then we measure that barium sulfide, or we measure the sulfur isotopes on the mass spec from that barium sulfate. So if you have other sulfate that's just floating around in that water, then that's going to contaminate your signal. And so that's why we do all these rinses to remove forms of sulfur that are not from carbonate-associated sulfate. But that's not all we have to do. We also need to think about preventing pyrite oxidation, because when we add acid, you could dissolve some pyrites, and that could contaminate your signal. Now, the good news is pyrite oxidation with acid is relatively slow, so we can do things like limiting acid exposure to minimize the effect of pyrite oxidation. Or there's other things that people do, like perhaps they perform it anoxically, so they do this under a stream of nitrogen. Or acidify with stannous chloride, which keeps iron reduced such that you don't have iron oxides that would oxidize your pyrites. And so when I was in grad school, it felt like every 2 years or something, there'd be a new paper about CAS procedure and pitfalls and best practices. And so there's a lot of different papers that you can refer to. I think what the community is sort of centering on is that the best practices are to do a series of rinses with sodium chloride, and do those rinses and then test the rinse water to see that you're progressively removing your sulfate. And then once you get to the point when your rinse water has no sulfate in it, then you can oxidize. Then you can acidify your sample. And so some people recommend that you perform it anoxically, and it's really important to limit your exposure to acid. And so this is just an example, this Wote paper. I'm sorry you shouldn't be able to read it. I just wanted to put it up there as an example. But you can read these papers, but I really do think that this is what our community is sort of centering in on. I do want to mention that you should beware. So a lot of sodium chloride that you get from like sigma or hydrogen sulfide has trace amounts of sulfate. So do your due diligence and do a bunch of blank analyses to make sure that you're not going to be contaminated, or that you don't get contamination from the sodium chloride. And I do think that you should test your methods before you start. So this is just an example from when I was a grad student. I took a bunch of representative samples from that had different characteristics that might make it harder to get a really robust sulfur isotope signal. So I chose some beige rocks, but then also some gray rocks that I interpreted to mean that they had higher organic matter or pyrite, or some red rocks, which I took to mean that they would have higher amounts of iron oxides. And I did a bunch of different method tests. And ultimately, I was able to choose the one that did the greatest job of removing non-sulfur, non-carbonate associated sulfate sulfur forms, as well as gave a consistent result. And so I encourage you to do that when you're starting. Okay, so what other considerations? So the other thing is you want to be careful in how you select your samples. So you should select carbonates that have minimal evidence for secondary carbonate formation. And you also want to sort of pick something, if you can, that's a single type of carbonate. And this is because different types of carbonates can have slightly different sulfur isotope compositions of CAS. And so this is sort of two examples. So on the left, we have a study from President et al., where they're able to microdrill a bunch of different alkyms, as well as micrite, and then do a bulk CAS analysis. And they showed that you did see a lot of variability between these different carbonate forms. And so choosing something like a brachiopod, which is a pretty large, robust shell that is resistant to alteration, can be really great. So if you're able to choose brachiopods, then I would just target that. Or, in the present study, they did show that micrite was pretty variable, but I've also gotten some CAS records from micrite rocks that are not that variable and show similar isotopic trends to other proxies. And so I do think that can be a robust recorder. However, doing some petrographic analyses to look at what that micrite is actually composed of, whether or not it's composed of cements, or if you can see evidence for different types of organisms, microorganisms within it, that can be really helpful. And the other reason why we want to choose a single type of carbonate, if we can, is that different types of carbonates can have different concentrations of carbonate-associated sulfate. So on the right, this is a color image, which is showing you concentrations of carbonate-associated sulfate in a sample. And you can see that there are certain places where there's a lot more CAS than others. And so if you have this sort of mixed sample that has a lot of different carbonate types, then your signal that you see will be biased by whichever carbonate type has the highest CAS concentration. So I don't think a lot of people are going out there and trying to measure Packstone sulfur isotope values by grinding it all up. But if you can, try to target samples that are single carbonate type that is usually robust to secondary carbonate formation. Okay, then we also need to think about diagenesis. And so sulfur isotopes, as well as concentrations of carbonate-associated sulfate, can be affected by post-depositional processes. And so I have two studies up here, which I think of as like the angel and the devil of carbonate diagenesis and how they affect carbonate-associated sulfate sulfur isotope values. So on the left, this study was from the Florida Keys, where they had some primary aragonites that were right next to a reaction center where the carbonate had been transformed to calcite. And so what they showed was that this transformation was associated with the loss of carbonate-associated sulfate. So carbonate-associated sulfate concentrations decrease. This is why we don't use carbonate-associated sulfate concentrations as proxy for marine sulfate. But despite that CAS loss, the sulfur isotope compositions of carbonate-associated sulfate retained the Pleistocene seawater sulfur isotope value. And so this was taken to show us that sulfate concentrations of CAS might decrease during alteration, but the sulfur isotope signature should be preserved. So this is good news. But on the right, we have an example from the Kleino core, which is one of our greatest sort of examples of how diagenetic processes can alter geochemical signals. And so here, sulfur isotope compositions of CAS are plotted in red, and this light red line is the sulfur isotope composition of seawater sulfate, and so that's what it should be if CAS is recording ambient seawater sulfate sulfur isotopes. And what you'll also see is there's this plot over here which shows us the different carbonate minerals. And so what they showed is that at this lower dolomization front, the upper part of it, which had evidence for hard grounds and other exposure surfaces, so those dolomites had values that were very similar to the sulfur isotope composition of seawater, and that's because this was a seawater-dominated diagenetic regime. Whereas at depth, it was likely that it wasn't seawater diagenesis, but some other form of diagenesis with really high water to rock ratio that was affected by marine pore waters, and therefore that did alter the sulfur isotope signal. So this should just show us that we need to be careful. This should give us a little bit of pause. There are processes that can alter the sulfur isotope signatures, but if we look at the lithology and use geological information, we might be able to discern which sections are more likely to preserve a primary signal versus which aren't. And then finally, we want to use geochemical tools to assess the fidelity of these records. So we're really lucky when it comes to CAS that the carbon and oxygen isotope people have been using cross plots for a long time in order to understand diagenesis and the alteration of carbon and oxygen isotope signals. So we can use a lot of the tools that people use to assess the fidelity of carbon and oxygen isotope records to assess the fidelity of carbonate-associated sulfate records. And so this is just an example, and I'm sorry that all the labels are so small, But what is done is that you plot your concentrations or isotopic compositions versus various indicators of diagenesis. And if the indicators of diagenesis are below a certain value and they don't show any variability, or if your isotopes don't show any variability as a result of them, then you're usually confident that you're recording a primary signal or something that hasn't been that affected by diagenesis. So this is another thing that you should do, is you should measure these other geochemical indicators to assess the fidelity of your records. And so I'm giving you a lot of information about the pitfalls and how things can go wrong, but I don't want you to, say sulfur isotopes aren't useful. These records are still very useful. And carbonate-associated sulfate is just one geological archive of information about sulfur isotope composition of marine sulfate. We can also use marine barite or sulfate evaporites, but they're often harder to extract or they're not that temporally continuous. So that's why carbonate-associated sulfate is measured so much. It's a lot easier to do, and we have a lot more of it. But the cool thing is that there are certain cases where we can see that these different archives of information about sulfur isotope compositions of marine sulfate, they actually preserve very similar patterns. And so on the left, I have an example from the Jurassic, where the carbonate-associated sulfate data in these smaller symbols follows very similar patterns to the evaporate data, which are in the larger signals. Of course, there are some large error bars, but for the most part, they overlap really well. And then there's another example from the Cretaceous where we can see that the sulfur isotope composition of CAS tracks this sulfur isotope record that we get from marine barite. Now, of course, they diverge at some point, and that might be due to the size of the sulfate reservoir. So this only really works if we assume that the reservoir, that the marine sulfate reservoir is really large, such that the residence time of marine sulfate is longer than the mixing time of the ocean. Whereas if sulfate levels are really, really low and the residence time of sulfate is much lower than the mixing time of the ocean, then we might expect there to be local variability in sulfate sulfur isotopes. So this can be something that gives us confidence in our records, and where possible, it is really useful to look at sulfur isotope records from multiple sites around the globe. And it's really exciting when they all line up and you feel like you're really measuring some robust signals. Okay, so now we're going to move on to pyrite. So pyrite is traditionally extracted as chromium reducible sulfur. So in some cases, yeah, people are going in there and micro drilling pyrite, but in a lot of cases that pyrite is finely disseminated throughout your sample. And so you have to extract it using various types of chemicals. And so we do this with a chromium reducible sulfur extraction. And so what happens is you take your sample, you put it in this vessel, get rid of all the oxygen by pumping it with nitrogen, and then you add in your chromium chloride solution, and that releases the sulfur from pyrite as hydrogen sulfide, and it is transported via nitrogen carrier gas into a trap vessel where it's trapped as zinc sulfide, and then it's transformed to silver sulfide, which is what we measure on our mass specs. And the solution is actually really beautiful. This is a chromium chloride solution. Now, when you buy the chromium chloride from Sigma or Fisher or whatever, it's oxidized, and so it's sort of like emerald color. But then you acidify it and react it with zinc, and it gets reduced, and it forms this beautiful, almost translucent blue color. And so this is what you want your solution to look at. Like, if you come into the lab and you have a dark blue solution that you can't really see through, you need to re-reduce it. Add some more zinc, add a little bit of acid, and you can get it back to this nice blue color. This is what you want. So an important caveat here is that this extracts all acid-soluble sulfides. So it will also recover iron monosulfides, metal sulfides, zero-valent sulfur. And so when we're working in modern sediments, we usually do an organic solvent extraction to get the zero-valent sulfur. We also do an acid volatile sulfide reaction to get the iron monosulfides. And it's only after we've removed those that we actually do the CRS extraction. When we're measuring rocks, sometimes we just do CRS extractions because we assume that iron monosulfides aren't persistent over long time periods, or that there are not a lot of metal sulfides. But sometimes there can be those things in there. So it is a good idea to run acid volatile sulfide extractions on some representative samples to see whether or not you have those phases and if you should worry about them. And this is not to talk about iron speciation, but I want to make the point that this also affects iron speciation data. So part of the iron speciation extraction is, or the way that we quantify iron as pyrite and iron speciation extractions is you do this CRS extraction. You measure the sulfide and you assume that for every 2 moles of sulfide, you'll have 1 mole of iron because pyrite is FeS2. But if you have iron monosulfides, then you're getting your iron concentrations wrong. So those of you who measure iron speciation, I want you to think about this. There are some folks who use nitric acid to leach pyrite. And so sometimes looking at the method section to see exactly what operational definition people are using could be useful. I also want to say that you should save your CRS residuals. What is contained in the stuff that's left over is organic sulfur. There can also be marine barite in there, but you can screen for that. But these organic sulfur isotopes can also provide extra information. And so I'm not going to go into it. There should be a whole nother primer proxy about organic sulfur, but it can be useful to measure organic sulfur isotopes in combination with pyrite to extract paleoenvironmental information. So photography is really important here because looking at your pyrites and can be really useful in your interpretations. And so I have an example here from some samples from the Draken Formation where I was measuring pyrite sulfur isotopes with secondary ion mass spectrometry. So individual grains, which is a really powerful tool, but usually, but it is very time intensive. So you can't do it on all your samples, but it is useful to look under petrographic scope, because in this case, what we could see is that there are 2 populations of pyrite grains. There are these small pyrite grains that are finely disseminated in organic rich ground mass. And then there are these larger pyrite grains that are primarily found on the boundary of void spaces. And using cross-cutting relationships, we can make the interpretation that the larger grains were formed later. And so we were able to show that the smaller grains, which generally had higher sulfur isotope values, or we were able to show that the smaller grains had higher sulfur isotope values and the larger grains had lower sulfur isotope values. And we interpreted this as indicating that the system was dominated by the metabolic differential effect, so the effect of sulfate reduction rates. So the smaller grains were formed near the surface where there was high rates of sulfate reduction and low fractionations, whereas these larger grains were formed later in a deeper environment that had lower rates of sulfate reduction, higher fractionations, and therefore lower sulfur isotope values. So whether or not you're able to do SIMS, it's still useful to think about what are the different, where are the pyrite grains found in your found in your sample, and then are there different grains that might be capturing signals from different time periods or different locations. And I want to make another point, which is that you should really use reflected light when you're looking for pyrite in your samples. Sulfides are not the only opaque minerals. Magnetite can be opaque. Organic matter sometimes shows as opaque. So if you really want to do a good job of identifying your pyrite grains, you should really use reflected light. And this is really important because if you don't have sulfur in your sample, you can't measure sulfur isotopes. And this is true also when we're thinking about sampling. And so this is a study showing that surface oxidation, modern oxidation, can affect pyrite sulfur isotope records. And so what and colleagues did was they took some samples from outcrop. They removed obviously weathered surfaces, but they had outcrop samples. But then they also had one of those portable drills, and so they're also able to drill cores. And what they showed is that the core tops and the outcrop samples had higher sulfur isotope compositions of pyrite than those in the core samples. And so you need to be worried about surface oxidation. One thing to do is to dig trenches. And so here's me as a grad student with a lot less gray hair, trenching in to try to get some really pristine samples. So that's one thing to do, is to make sure that you're digging in as far as you can. But then also another good idea is to measure from core samples because those have been exposed to the atmosphere for a lot less time. And I also want to make the point that this also affects ion speciation as well, because usually the pyrite oxidation will decrease the amount of sulfur in your samples, and therefore you might be underestimating the pyrite in your sample. So this is a really great study that I encourage everybody to look at. Yes, there must be sulfur in your sample in order to measure sulfur isotopes. So it's really a good idea to avoid CIDR samples with obvious evidence for oxidation. So things like red iron oxides is something that should give you pause. Okay, so zooming out a little bit, we also need to consider that pyrite is affected by local diagenetic processes because pyrite, is forming in the zone of sulfate reduction. So it really is recording local signals. And so what we can see is that along a deep to shallow gradient, we do see variation in sulfur isotope compositions of pyrite. And so in deep cores, in this example, we tend to find relatively low sulfur isotope compositions. And that's because, well, there's a few reasons for that, but one of the hypotheses is that you have low sedimentation rates, which favors open system conditions, such that you're able to preserve sulfur isotope compositions of pyrite that are relatively low. In contrast, in shallow sections, you have higher sedimentation rates and these reworking events that can close off your zone of sulfate reduction from the overlying sulfate reservoir, and those closed system conditions favor the preservation of sulfur isotopes that are higher. Aller et al., this is a really great study where they're looking at these mobile mud belts where sediment reworking reoxidized pyrite, resulting in relatively high pyrite sulfur isotope values at the boundary between this reoxidized sediment and the underlying methanic sediment. So that can also play a role in the fact that these sulfur isotope compositions of shallow systems are relatively higher, but also more variable. Now, the reason why this matters is because of Walther's law, right? So Walther's law is that environments that are next to one another today are stacked on top of one another in the geological record. So if we stack these records, we can essentially make a sulfur isotope excursion without there being any change in global sulfur cycling. And so if we go from high sea level, where we're in a deep environment, to low sea level, and then back to high again, then we might predict that we would see these changes, which again might be telling us about the evolution of our environment and depositional environments, but probably not telling us about changes in global biogeochemical cycling. And this has been shown. So this is a study from the Gulf of Lion, where they showed that during interglacial periods, when it was a deeper water environment, you had low sulfur isotope values, whereas during glacial periods, sea level was lower, and so sulfur isotope values were higher, likely because of higher sedimentation rates, but also it could have to do with organic matter. So the organic matter in nearshore environments is more easily degradable, and so therefore you might have higher fractionations, higher rates of sulfate reduction and lower fractionations, which would drive your sulfur isotope values higher. And so it's probably a combination of both these things, but really what we get out of this is that sea level can affect the sulfur isotope composition of pyrite. And this is something that we need to think about when we're doing these interpretations. And it seems like this actually is getting us towards understanding something that is interesting about global biogeochemical cycling. And so this is a study that shows that for the last 200 million years or so, the sulfur isotope difference between sulfate and sulfide that's preserved in the geological record is correlated with shallow shelf environments. And so I think this is something that people are coming to terms with, which is that the proportion of shallow shelf environments where you have high rates of pyrite burial, as that changes, that can impact the total pyrite burial flux. And so this can relate back to our box models, because it could just be these changes. One of the ways that we could change pyrite burial rates is by changing sea level. And so that's another way that we might think about our sulfur isotope records. So given all that I've said, I've talked to you a lot about the pitfalls and the issues and things that we need to think about when we're interpreting records. Are these records still useful? I'm biased, of course. I think yes, I love sulfur isotopes, but I think that we really need to be careful about how we're interpreting them. So if we want to get towards this box model interpretation of sulfur isotope records, we need to spend a lot of time ensuring that these are primary signals, but then also thinking about the different processes that might increase pyrite burial or change fractionation that maybe aren't just changes in areal extent of anoxic systems, but other changes to the Earth system that might drive those changes. So I'm going to just leave you with this list of things to consider when interpreting sulfur isotope records. So I'm not going to tell you how to interpret your sulfur isotope records, that's your job, but I want to give you a lot of things to think about. Because these are different things that affect your records, and I want them to be things that are in the back of your mind when you're thinking about sulfur isotopes. So one is global biogeochemical cycling, and these connections with the oxygen and carbon cycle, and how changes in the areal extent of anoxia or sulfidic systems might impact sulfur isotope records, or things like volcanism, weathering, evaporite burial oxygenation. But within that, we also need to think about open versus closed system sulfur cycling, accumulation rates, and sea level. We also need to think about sulfate concentration, the size of the sulfate reservoir, how sulfate reduction rates might change. And then we need to think about, are we recording local versus global signals? How is diagenesis affecting our geochemical records? And then what sort of contamination do we have during extraction? So I really enjoyed talking about this, and I really look forward to the question and answer session. Thank you very much.
Great. Well, thank you so much, Maya. That was really awesome. So we're now going to turn the, everyone can thank her for an awesome talk. We're now going to turn the video off now, if I can remember how to do that. There it is. And people will be able to ask kind of basically any questions that we want. And of course, we want to encourage people, you know, if you're not a geochemist, you are the target audience, or if you're not a sulfur biogeochemist, you are the target audience. So we want, you know, this to be an educational opportunity for the group. So
so it does still say you're recording. You might need to try it.
Yeah, I'm stopping right now.
Okay, cool. There you go.